Hello, I'm Austin Kim, and I'm basically going to go over the processes of mitosis and meiosis. And considering this project is mainly centralized along the intricacies of meiosis, I will first be giving a brief overview over mitosis and then get into the meatier details of meiosis in great detail. Now, just before we move on to the slides, which will bring you up to speed on meiosis and mitosis, basically, the cell cycle is composed of a variety of phases. First is G1 phase, which is the growth phase, the S1 phase, G2 phase, which are all parts of interphase, by the way, and then comes the M phase, which basically is inclusive of mitosis and cytokinesis. Now, these stages of the cell cycle can also be considered checkpoints as well. And both meiosis and mitosis are collectively very important because the chromosomes that are produced can be used in a variety of ways, whether to create new organisms through means of sexual reproduction, utilizing circular chromosomes to help create more DNA. They can be used in a variety of ways. And in addition to these variety of ways, one of the main key differences between meiosis and mitosis would be that meiosis is inclusive of sexual reproduction and creating individualized, unique organisms and cells, while mitosis is quite literally coherent to asexual reproduction and binary fission and creating an identical replication of the original father cell. Now let's actually go on to mitosis after this basically a summary / introduction of what this movie is going to be about. Now we move on to the process of cell division known as mitosis. Mitosis is broken up into 4 substages known as prophase, metaphase, anaphase, and telophase. Prophase is notably the first and longest phase of mitosis. First of all, the centrioles separate and take up positions on opposite sides of the nucleus, likewise. Now, this is just a rough drawing, and please excuse my poor artistic skills. Some people are born with certain gifts. I was not born with the gift of drawing. Now, there is also the presence of a centromere right around this general vicinity, and within it are just a compilation of different chromosomes, which are basically just paired chromatids, likewise, and there will be multiple amounts. These are just some examples. In this phase, the nuclear envelope breaks down and chromatin condenses into chromosomes, as you can see right over here. And there are also some spindle fibers that form as the centrioles separate in this vicinity as well, and that basically wraps up prophase. Now, the second step of mitosis, or second stage rather, is metaphase. And in this phase, the chromosomes purely line up across the middle of the cell. And here, microtubules connect to the centromeres of each chromosome and connect them to the poles of each spindle. Just like this. Next is the third phase of mitosis, which is known as anaphase, which is also regarded as one of the shortest phases of mitosis as well. Here, the sister chromatids actually separate into individual chromosomes, and the 2 pairs of individual chromosomes now separate and move in opposite directions until they form 2 distinct groups, and spindle fibers help pull them apart from each other and create this individualizing process and make it even easier. This is a very rough drawing of what it might look like. Now, here is indicative of the final stage of mitosis, which is known as telophase. Here, chromosomes gather at opposite ends of the cell and lose their distinctive shape as they have given up 1 pair of chromatids and it is equally distributed to 2 different cells. To ultimately seal the deal on this new process, a new nuclear envelope clusters around each group of chromosomes, as you can see here and here, which ultimately forms a new cell, which is a directly identical replicate of the prior cell. And lastly, as a result of mitosis, normal human somatic cells have 46 chromosomes. And lastly, before moving on to meiosis, there are a variety of both internal and external regulators that can control the rate of mitosis and cell division. Cyclins, which are also part of MPFs or maturation promoting factors, can greatly decide the timing and rate of the cell cycle, especially in cell division. So basically, we move on to the next process of meiosis. Meiosis inherently is basically a process of reduction division in which the number of chromosomes per cell is divided in half through the separation of homologous chromosomes in a diploid cell. Now, this process is pretty similar to mitosis, has similar phases and substages such as prophase, metaphase, anaphase, and telophase, except there are two stages of actual meiosis, and rather than mitosis, when a direct replicative cell is created, meiosis actually introduces diversity by mixing chromosomes through a process called crossing over or synapsis, which I will go on to explain more in the next slide. The first step is meiosis 1. Now, the first substage of meiosis 1 is interphase. Now, in this stage, cells undergo a round of cell duplication. This creates duplicate chromosomes. So first we deal with the first stage of meiosis 1, which is prophase 1. Now, what basically happened in this specific substage is that each chromosome pairs with its corresponding homologous chromosome to form a tetrad, and there are 4 chromatids in an actual tetrad as well. You can see that the centriole pairs are on opposite sides, just like this. So when homologous chromosomes form tetrads in prophase 1, they exchange portions of their chromatids in a process called crossing over, which can also be called synapsis. Now, crossing over or synapsis, as you can see here, exchanges perhaps one chromatid and another chromatid of another chromosome, and they interchange and exchange them. And this can produce new combinations of alleles, which can actually introduce diversity and independent assortment at the same time, which is pretty cool. The next stage of meiosis 1 is metaphase 1, which basically is similar to the metaphase of mitosis, as the chromosomes line up in the middle of the cell and spindle fibers connect them to the opposite poles of the spindle using microtubules. The only difference is the chromosomes that are lining up in the middle are now recombinant chromosomes, which are independently assorted as they each have individual parts from the other chromosome that they originally got through synapsis. And basically, the microtubule attaches to the kinetochore, and the centromere, along with the kinetochore, is then connected to each side of the spindle. And this is essentially the process in which the tetrads line up. So here now is anaphase 1 of meiosis 1. This is extremely similar to anaphase of the mitosis variety as well. In this one, instead of normal chromosomes moving to opposite poles of the cell and being pulled by spindle fibers, this time the homologous chromosomes are being split apart and are moving to the opposite poles of the cell. So now is the final stage of meiosis 1. This is called telophase 1, and cytokinesis is also a part of it. Now basically what happens is telophase 1 can initiate when the chromosomes in their 2 separated groups can actually reach the poles. Then the nuclear membranes eventually begin to form after the spindle breaks down, and the nuclear membranes form on account of cytokinesis, which basically splits the cytoplasm. The cell then separates into 2 separate cells, and the 2 cells produced by meiosis 1 have chromosomes and alleles that are completely different from each other and from the diploid cell that had entered meiosis 1 in the first place. These 2 new cells can be dubbed as 2 haploid daughter cells, which have duplicated chromosomes and evidently appear after cytokinesis has completely finished taking its course. Now, the two cells produced by meiosis 1 undergo a second meiotic division. One of the key differences that will be apparent in meiosis 2 is that neither of the cells go through a chromosome duplication. Each of the cell's chromosomes contain two chromatids as well. So, as stated before, meiosis 1 results in two haploid daughter cells that are made, each with half the number of chromosomes as the original cell, as you can clearly see over here. This is basically what is evident in prophase 2, which basically just establishes and shows the two haploid cells with half the number of chromosomes. Now in my metaphase 2, the chromosomes, or homologous chromosomes, simply line up in the center of the cell once again. Now in anaphase 2, the sister chromatids, is shown here, actually separate once again and move towards opposite ends of the cell, which is extremely similar to what happens both in meiosis 1 and just mitosis in itself. However, the result of meiosis 2 is 4 haploid daughter cells. And in the example shown right here, each of the 4 daughter cells produced in meiosis 2 receives 2 chromosomes. These 4 daughter cells now contain the haploid number, or N, which is just 2 chromosomes each. Now, as it will be further explained in the next slide, the cells that will develop into sperm or egg cells that will be ready for sexual reproduction are called germ cells, or germline cells actually. Now, in terms of gamete formation, it differs with the gender in which the formation is taking place in. However, in males, there are 4 equally sized gametes, which are called sperm, and the process in which these sperm gametes are produced are called spermatogenesis. Also, in more specific terms, a male gamete can also be referred to as a spermatocyte. Now, when speaking about oogenesis, it is not too different from the formulation of sperm. However, in many female animals, only one egg actually results from meiosis. The other 3 cells, which can be portrayed here, which are called polar bodies, are generally not involved in reproduction. Because only one egg is actually realistically going to be fertilized by the countless sperm cells, or whatever sperm cell first reaches the egg, in sexual reproduction, the rest are simply discarded or simply recycled by the lysosomes in the cell. And the prime reason why genetic diversity can be introduced to new organisms is all tied back to the original synapses and crossing over processes that occurred first in meiosis 1, which allow certain individual organisms to be different from others when they come out, maybe giving them a certain advantage in their environment, allowing them to top the competition, to survive and be naturally selected while other organisms may die out. This is all how genetic diversity and independent assortment is basically implemented. And that concludes my presentation. So thank you so much for listening.